Now see that basically the two models have been proposed to explain the linkage between that polyadenylation and the termination. So transfer of 3' processing enzymes from the polymerase that it will trigger the conformational change. या ठिकाणी दोन मॉडेल दिलेल्या still it is not known that which will determine the length of the adenine residue and how this particular ending process, that cleavage from that RNA polymerase, that RNA will be removed from the RNA polymerase, RNA polymerase will remove from that DNA. Still this process is not fully known, but there are certain hypotheses. There is a connection or relation between the polyadenylation and the termination. So first one is that transfer of 3' processing enzymes from the polymerase, it will trigger RNA into the conformational change in RNA polymerase, and which will reduce the processability of the enzyme, leading to a spontaneous termination. I will repeat this point once again, that the transfer of 3' processing enzymes. Okay, 3' जो एंड होता आरएनएचा त्या ठिकाणी जे की प्रोसेसिंग एन्झाइम्स होते पर्टिक्युलरली विच आर रिक्रुटेड बाय दी सीपीएसएफ अँड सीएसपीएफ दोन फॅक्टर आपण बघितले. सो दिस फॅक्टर्स CTSF and CPSF, these factors will recruit the number of enzymes, and these enzymes which are present at the 3' end, these enzymes which will trigger the RNA structure, and RNA जर cleave झाला, it means that this is the signal for the RNA polymerase to stop that activity, means to terminate that transcription. Then the next one is that the second model proposes that the 5' cap on the second RNA molecule is sensed by the polymerase, and which recognizes the transcript as the improper and terminates. According to two models, it is given in the Watson book. So according to the second model, it says that the capping itself, it is responsible for the polyadenylation. The absence of cap reflects the capping enzyme on the RNA molecule. It is this stage of the transcription cycle. We will not waste our time on this particular two models, but this, according to this model, that polyadenylation and that splicing, these are interconnected with each other, and somewhere that the termination of that transcription is also connected with this enzymatic activity. Now the transcription termination, once the RNA polymerase reaches up to the end region of that particular gene, it will encounter certain specific sequences that after being transcribed into the RNA, and that these sequences will trigger the transfer of the polyadenylation factors or that particular enzymes to the RNA. As already I told you that transcription termination, two factors, that was CPSF and CSTF, these two factors, they are responsible for the termination of that transcription was going on there, because these factors are responsible for the cleavage of that RNA from the RNA polymerase, and without the RNA, that RNA polymerase will not work over that gene. It is said according to that particular model. That's why the RNA polymerase very soon will release that DNA, and obviously indirectly this process is called as the termination. So once the RNA polymerase reaches up to the end part of the gene, it will encounter specific sequences. These are called the poly A sequences. If you remember that here at this region. So once the RNA polymerase will reach up to these sequences, after that these sequences will be added into that RNA, and once these sequences added into the RNA, this factor, that is CSTF and CPSF, this factor will be recruited in that particular area, and it means that indirectly it is the termination of that transcription process. Then leading to the three events. When there is a termination of the transcription, at that time there will be three events will occur. Cleavage of the message, that is RNA. So RNA will cleave from that RNA polymerase. Then second thing is that polyadenylation. So many of the adenine residues will be added. Near about 200 adenine residues will be added by the poly A polymerase, this enzyme. And the third one is that transcription termination, which was going on by the RNA polymerase. So RNA polymerase will also leave from that site, from the terminator sequences. So in this way, this was the termination of that transcription. Now we reach up to the point that till now the RNA is successfully formed, right? Now see here that the next thing is the unwanted part of that RNA, that is called as the introns, it should be removed from that RNA. That is called as the splicing mechanism. So one by one we have to learn here the splicing. So eukaryotic genes are consist of the blocks of coding sequences that is called as the exon, which are separated alternately by the other sequences which are the non-coding sequences called as the introns. So if you see in this particular diagram, this is the pre-mRNA. Now see, this is the genomic DNA. The gene itself contains the coding sequences and the non-coding sequences, or coding part and the non-coding part. Then this gene will be transcribed. Now here is the pre-mRNA, which again contains this one, that is exon and the intron part. Now during the splicing, what happens that the unwanted part or the non-coding part, non-coding region, that is intron, it will be removed out, and only the coding part, that is exon, will be joined, or ligation will be there. Now it is called as the spliced mRNA, or it is also called as the mature mRNA. This mRNA is now ready for the translation. Still this process goes on or occur within the nucleus or inside of the nucleus. After this splicing, total splicing mechanism, the mRNA will be transported outside of the nucleus. It will reach into the cytoplasm, and where the ribosome will read these codons and it will form the chain of the amino acid. Now, the introns are removed from the pre-mRNA. This is the pre-mRNA. The introns are removed from the pre-mRNA, and the process called the splicing. And this process converts the pre-mRNA into the mature mRNA. The lack of accuracy in this splicing mechanism. If there is a lack of accuracy during the splicing mechanism, what will happen that either the important or necessary base will be lost or gained. Either it may happen that the necessary base will be removed or lost, or unnecessary base may be added or included or the gain during this splicing. That's why the open reading frame, last time I have explained that whatever the in between the 5' UTR and 3' UTR region was there, it is called as the open reading frame, which will be read by the ribosomes for the formation of RNA. That open reading frame will contain the unusual bases. And obviously, if there is a faulty splicing, it means that it will be responsible for the production of the faulty protein. Then, molecular mechanism, we have to learn here the splicing at molecular mechanism though. So the molecular mechanism of the splicing occurs through specific sequences which occurs within the pre-mRNA. So I repeat again that because this slide will not repeat again and again. So see here, this is the pre-mRNA and this is the mature mRNA, or the mRNA which is spliced mRNA. So wherever I will use the word that is pre-mRNA, pre-mRNA means what? The mRNA which contains the exon and the introns. And mature mRNA means what? The mRNA which from which that introns is removed or spliced out. Here again, you see here that this is in this particular picture, only the part of the intron is shown. This is the intron. See here, this is the intron, and this is the part of exon. So two exons are there. And in between the two exons, there is an intron, and this intron should be removed from this area. So who is responsible for this removal of the intron? We will see here one by one at molecular level. So first of all, see here that this is the part of intron. This is the area which is called as the 5' splice site, and this is the 3' splice site. And always it contains the adenine. So it is the conserved. Then the 5' end of the intron is marked by sequences called as the 5' splice site, and the 3' end of that intron is marked by some special sequences which are conserved here, known as the 3' splice site. Always there will be the adenine and guanine. Exon will end by the guanine residue, and intron will start with guanine, then uracil. The third position may contain either adenine or the guanine, and then fourth one adenine, guanine, and uracil, and so on. In this way, the nitrogen bases will be here, and at central region, particularly not exactly at central, that this part is very little bit somewhat closer to the 3' splice site, and this region, this particular area contains the adenine. This is called as the branch site, and this one again 3' splice site of the intron. Here there may be the pyrimidine residue may present here, either cytosine or the uracil, and the last one, that is adenine and guanine. Again, the exon will start with the guanine. So one by one, we will learn here about all these things. So the third sequence necessary for splicing is called as the branch point. For the splicing, 5' splice site, 3' splice site and branch site is responsible or necessary. Then the most highly conserved sequences, because last time I have explained the difference between the conserved and the consensus sequences. So this is the conserved sequences which are reached in GU at this 5' splice site, and in the 3' splice site, and A at the branch site. Whatever the residue we can observe here, all these are the conserved sequences along with this A also. Splicing mechanism or splicing is achieved by two successive trans-esterification reaction in which phosphodiester linkages within the pre-mRNA are broken down, and again they are formed. So this splicing mechanism occurred by the two successive trans-esterification. So how it will carried out, we will see here. Two times trans-esterification reaction will be carried out at this same place, at this area where the intron is present. How it will occur, we will see one by one. That during the first reaction is triggered by the 2' OH of conserved adenine at the branch point. This group acts as a nucleophile to attack the phosphoryl group of the conserved G in the 5' splice site. So see here in this diagram, you will get the clear idea that this is the branch point, right? This is the branch point where that adenine is present. Adenine will be always present in that branch point because this is the conserved sequences. 2' OH of this adenine will attack over the conserved guanine, which is present in the 5' splice site. We know that in between this guanine, then uracil, see here, uracil, guanine, and adenine. In between that, these bases are joined by the phosphodiester linkage. So to understand in a better way, they have shown that there are phosphate groups. It indicates that there is a phosphodiester bond is present in between these bases. So this 2' OH, this oxygen, it acts as a nucleophile. It will attack over this phosphodiester bond in between these, particularly where the guanine, which is present in a conserved region of the 5' splice site. Thus, the phosphodiester bond in between these two sugar molecules will be broken down, and one junction will be formed in between the intron and the exon, and the free 5' end of the intron is joined to A, or that is the branch point within the intron. See here that what is going on here. After attacking over this phosphodiester bond, now the 3' OH of this exon will remain open. The phosphodiester bond in between the ribose sugar is broken down because of this attack of the oxygen. Now it will open. Now this is the exon. Exon contains the 3' OH. It will fold in such a way that it will form the structure like this. Okay, so this is the lariat structure like a lariat. So commonly it is called as the lariat. Now, next step is that in addition to the 5' and 3' backbone of the linkages, a third phosphodiester extends from 2' OH of that A to create three-way junction, hence known as the branch point. So here, this oxygen acts as a nucleophile. It will attack over this exon part. So at that time, it will join, this exon will join with this other exon. So during the joining, what will happen that it will cleave this bond, which is present in between that, or the phosphodiester bond, which is present in between that, this intron and the exon. So in this way, intron will be removed and two exon will be joined. So 5' exon is leaving group in first trans-esterification reaction. Okay, now see, in first, because 2 times the trans-esterification reaction is occurred here, so you will get confused in between the first sentence and the last one. 5' exon is leaving group in first trans-esterification reaction. In second reaction, 5' exon reverses its role and becomes the nucleophile that attack the phosphoryl group of the 3' splice site of that particular intron. So this second reaction has two consequences. Means two results will be there. The first and most importantly, it joins 5' and 3' exons. Now see that this oxygen will act as a, or particularly you can say that it's the exon. So this exon, this is the 5' site of that particular exon. So this was nothing but it will attack over the 3' splice site here, and it will break this phosphodiester bond. So after breaking this phosphodiester bond, it will join, this will join with this other exon. In this way, these are the joined two exons, and at the same time, this intron will be removed. So intron lariat, this intron or lariat intron will be removed or released during this. So actually, this is the splicing mechanism. In second reaction, two successive consequences, that is first and most importantly, it joins 5' and 3' exons. Hence, it is the step in which the coding sequences are actually joined or spliced together, and in second step, it will liberate or remove or release the introns which serve as the leaving group. So that's why I already told you that in first there was 5' exon was leaving group, and in second there was intron act as a or serve as a leaving group. So introns will be leave from this area. That's why it is called the leaving group. Then in the two reaction steps, there is no net gain in the number of chemical bonds. Whatever the two transcriptional reaction we have learned here, that in these two reaction steps, there is no net gain in the number of chemical bonds. Two phosphodiester bonds are broken down and two new phosphodiester bonds are synthesized. So there is no any net gain. Then transesterification reaction is mediated by huge molecular machinery called as the spliceosome. Though in that particular diagram, in the previous diagram, I have not shown any factor or the spliceosome machinery or the proteins, but actually behind this mechanism, number of proteins or the factors or the machinery it is required, it is given in the next slide. We will learn again that one by one. That is called as the spliceosome. Zome is the body, and spliceosome means the body which will carry out the or which is responsible for the splicing mechanism. So obviously in earlier lectures also I told you that within the cell, whatever the activity occurs, all these activities done by the factors, and factors are nothing but always these are the proteins. This spliceosome machinery composed of near about 150 proteins and 5 rRNA, and which are similar to the ribosomes. So many of the functions of the spliceosome are carried by these components than the proteins. Thus, the RNA locate the sequences, the elements at the intron exon borders and participate in the catalysis or to break down that bond. So one by one, you have to just remember behind the splicing, certain factors or the proteins are responsible. So how many proteins are there? There are approximately near about 150 proteins are present. Along with that, 5 rRNA molecules. 5 rRNA we will designate as U1, U2, U4, U5, and U6. Okay. So all these are the small 5 RNA molecules. So these RNAs, they are collectively called as the nuclear RNA or the small nuclear RNA or the snRNA. I repeat again that, see the last point there, the 5 RNA, which one? That is second point, what I told you that the transesterification reaction, whatever the reaction, the two times we have seen that the breakage of that phosphodiester bond and the joining of that phosphodiester bond. It means that simply the removal of the introns and the joining of exons. All it is nothing but it was done through the transesterification reaction, and this transesterification reaction is carried out with the help of the spliceosome. This spliceosome composed of the 150 proteins and 5 rRNA. Now this rRNA designated as the U1, U2, U4, U5, and the U6. And collectively, these are called the small nuclear RNA. And these RNA near about 100 to 300 nucleotide long. And these RNAs complexed with the different proteins. And at the time, we will call that this RNA and the protein, it is called as the snRNA. It means small nuclear RNA along with the protein complexes. So these snRNPs have 3 roles in splicing. So whatever the snRNP, snRNP means what? The RNA and the protein molecules. So this complex, that this SN small nuclear RNA and the protein complexes, they have the 3 roles in splicing. They recognize the 5' splice site and the branch site, and they bring those sites together as required and catalyze the RNA cleavage and join the reactions. Then, to perform these functions, that is RNA-RNA, RNA-protein, and protein-protein interactions, it is very important. It is not easy to interact any protein to interact with the DNA or the RNA. So that protein must contain certain structure, that is helix turn, helix motif, or the zinc finger motif, and so on. Here, the second point, you have to just remember that to perform this interaction, which interaction? That RNA and RNA is interacting. Now, which RNA is interacting with which RNA? That the intron, it is interacting with intron itself, or you may say that exon is joining with the exon, or you may say that intron is removed from the exon. So that is RNA-RNA interaction. Then RNA-protein interaction. RNA-protein interaction is what? The mRNA interacting with the proteins, and again, these 150 proteins interact with each other. So this interaction is called as the protein-protein interactions. So in first step in spliceosome assembly is binding of U1 snRNP, the particular 5' splice site of the pre-mRNA. U1 snRNP will attach or will act with the particular 5' splice site of the pre-mRNA. Then U2 snRNP then binds to the branch point. We know that branch point always contains the adenine because it is conserved, highly conserved. Then preformed complex of U4, U6, and U5. It means that these, particularly all these RNA will form the complex. It is called as the preformed complex. Either it may be the U6 and U5 is then incorporated in that spliceosome, and with U5 RNA, it will bind to the sequences upstream of that particular splice site region or the area. Then U6 dissociate from the U4 and displaces the U1 at 5' splice site. Then the whole reaction, whatever the author here will learn in this way. So here, this one, that was U1. Okay, so U1 and then U2. So U1 was attached in that 5' splice site, and then U2, it was present in that particularly branch point, and the remaining RNA, that is say U4 or U6, all they will assemble and they will carry out the complex that is called the spliceosome complex or the machinery. After that, U1 and U4 will be removed, and the remaining one, U1 and U4 is removed from that area, and who is remaining? U2, U5, and U6. So U2, U5, and U6 will remain in that particular area, which will be responsible for the formation of layered structure. It will dissociate, it will cleave that intron from that particular exon, and again, the remaining splices of machinery will be removed or released from that area. So in this way, indirectly, the splices of machinery, I have not explained this part in a previous slide. In previous slide, I have just explained according to the Watson that what is going on here through the trans-esterification reaction. See here that I said that this 2' OH of the adenine residue of that particular branch site will attack over the 5' splice site. But remember that along with this splicing machinery or the spliceosome machinery is responsible behind this or for this splicing. So just remember the role of that particular spliceosome machinery along with this RNA, that is U1, U2, U4, U5, and the U6, along with that 150 proteins. So obviously the remaining proteins, they are not shown here. Then the catalytic role of RNA in splicing was demonstrated by the discovery that RNAs are capable of self-splicing. Now we know that within the RNA world, that RNA may carry out the splicing without the help of any protein, because when there was protein was not present over the ocean or the earth, at that time RNA was splicing itself, and at that time it is called as the self-splicing. Now the catalytic role of RNAs in splicing was demonstrated by the discovery that some RNAs have the ability to self-splicing or self-splice out. So they can catalyze the removal of their own introns in the absence of the other proteins of the RNA factors. The meaning of this first point is that there are certain RNAs may be present along with these RNA, because in previous example, what we have learned, in previous slide, we have learned that these RNA were taking the help of the proteins, that is spliceosome, for the splicing mechanism. But we know that there are certain RNA molecules are there. These RNA molecules will cleave or will carry out this splicing without the taking help of any protein or any factors. Okay. So it was first discovered in tetrahymena. So it was first discovered during the studies of 28S RNA of the protozoa. One protozoa is there, that is tetrahymena. So additional studies was shown or showed that self-splicing RNAs in mitochondria and chloroplast and the bacteria also. So these self-splicing RNAs are divided into the two classes. Previous slide, I have explained that how the RNA was shown the splicing with the help of the certain protein molecules. means either RNA and the proteins, and that complex is called as the spliceosome. Okay. In this slide, we are learning that the RNA itself, they are cleaving, they are cleaving itself without taking the help of any protein or the factor. So this self-splicing RNA, they are divided into the two classes or the two groups on their reaction mechanism, that is group 1 and group 2. Okay. So many of the times you have heard in your lectures also, routine lectures or the regular lectures, there is group 1 introns and the group 2 introns, or the group 1 and group 2 RNA. One by one we will learn here that the first step in splicing for group 1 introns is cleavage at the 5' splicing site mediated by the guanosine cofactor. Remember the previous slide in which I have shown the branch point. In that branch point, there was the adenine residue, which was highly conserved, because each and every time you was observing that adenine is present. But here, there is a guanosine is a cofactor. Here, instead of adenine, in next slide, I have shown that particularly the guanine or guanosine cofactor itself, it will help during the cleavage. So 3' end of the free exon then react with 3' splice site to excite or to remove the introns as a linear RNA. See here that in previous slide that the RNA was removed in a lariat form. So self-splicing reactions of group 2 introns closely resemble. You will observe the same process within group 2 introns also. But how it will occur, how it will be carried out, we will see one by one here. That see carefully that those characteristics of nuclear pre-mRNA splicing in which cleavage of the 5' splice site result from the attack by adenosine nucleotide in the introns. So you are getting here the two diagrams over your left side and the right side. Whatever the diagram is shown on the left side, in which that particularly it is the group 1 intron. Okay. See here that this is the exon 1 and this is exon 2. This part is the 5' splice site, and this region is the 3' splice site. So this will be the 3' of that exon, and this will be the 5' end of that intron. Again, it will be the 3' end of that intron and 5' end of that exon. So do not remember in this way. Particularly, you have to concentrate. You have to focus only on the what intron. See here that this is the 5' splice site of the intron, and this is the 3' splice site of the intron. Now, when in case of the group 1 introns, guanosine, I already explained that guanosine cofactor is present. So this guanosine cofactor attacks the 5' splice site. So whatever the guanosine, which is the conserved, which is present in that particular area, where it is present in that 5' splice site. So this guanosine will attack the 5' splice site, and it will cleave that phosphodiester bond. And then whatever the cleavage of 3' splice site, ligation of exons. This one will be the 3' OH part, or oxygen will be there, and again it will be ligated along with that RNA and the protein molecules with the exon 2. So these two exon will be ligated, and this intron will be removed, but not in a lariat form. In other case, wherever there is a adenine is present or adenosine is present, so adenosine intron attacks the 5' splice site. We already learn about this whole mechanism, and then it will form the lariat structure, and then this exon will attach over this exon 2. It will cleave this bond or phosphodiester bond or the linkage, and then lariat intron will be removed, and two exon will be joined